Hello and welcome back to the Deep Dive. Today we're doing something a little bit different.
Yeah, we're taking a pretty literal journey today.
Right, into the machine that keeps you alive. We are diving into human physiology, specifically Chapter 16, Digestion and Absorption.
Which is a huge one.
It is, but listen, if you are a Neet aspirant listening to this right now, I need you to put down the highlighter for just a second.
Absolutely. Because the biggest mistake I see students making with this chapter is treating it like it's just a grocery list.
Yeah, exactly. Like salivary amylase, check. Pyloric sphincter, check.
Right. And if you do that, you're going to get tripped up on the application questions.
So we are going to completely flip that script today. We aren't just reading the textbook at you. We want to build a mental model. We're going to look at the actual logic of the body.
I love that phrasing, the logic of the body. And it ties directly into the introduction of Unit 5 in your source material.
The section everyone skips.
Exactly, the part everyone skips. But it's so important because it talks about reductionism versus systems biology.
Okay, let's unpack that really quickly. Reductionism. That's basically the classic scientific method, right? Breaking a clock down into its individual gears to see how it works.
Precisely. Historically, biology was just obsessed with taking things apart, these physicochemical concepts. The idea was, let's isolate this single enzyme in a test tube and see what it does.
Which is useful.
Oh, totally useful. It gave us molecular biology. It gave us the parts list.
But a pile of gears on a table isn't a ticking clock.
Exactly. And a bag of isolated enzymes isn't a living human being. The text really emphasizes this concept that life is an emergent property.
An emergent property, so the whole is greater than the sum of its parts.
Right. Digestion isn't just chemical bonds breaking in a vacuum. It is a massive, coordinated symphony.
Because you have the neural network signaling the hunger in the first place.
And the hormonal system prepping the acid.
And the muscular system actually churning the bag.
Exactly. So the mission for our deep dive today is to see that symphony. Not just the isolated instruments. We want to trace the journey of, say, a sandwich, from the very moment it hits your teeth until it literally becomes you.
That is a fantastic goal. And to keep this structured for your prep, we are going to break this journey down into 4 main phases.
Sounds good.
First, we'll look at the hardware, which is the anatomical tube, then the chemistry set, meaning the enzymes on the glands.
Then the logistics.
Right, the logistics of how we actually absorb all those nutrients, and finally, the glitches. The disorders of the system.
It's a solid roadmap. Let's get into it.
Let's start with the hardware. The alimentary canal. It basically begins anteriorly with the mouth, and it opens out posteriorly through the anus. It's a single tube.
A very complex tube. Let's zoom right into the very beginning, the oral cavity.
This is where mechanical digestion starts, and there are 3 very specific anatomical terms regarding teeth that show up on exams constantly.
You absolutely need to know these 3. The cotodont, Diphyodont, and heterodont.
Okay, let's break these down so no one has to memorize them blindly. The secodont first. This refers to how the tooth is actually attached, right?
Yes. In humans, each individual tooth is embedded in a dedicated socket of the jawbone.
Like a plug-in or wall socket.
Exactly like that. That specific type of rigid embedded attachment is called the secodont. It's not just glued to the surface of the bone like in some animals. It is rooted deep down.
The sicko kind of sounds like thick or anchored. That helps. Okay, next up is diphyodont. I'm assuming D just means 2.
You've got it. Most mammals, humans included, form exactly 2 sets of teeth during their life.
The baby teeth and the adult teeth.
Right. Technically the temporary milk or deciduous teeth, which are eventually replaced by the permanent or adult teeth. That conceptual framework of having 2 sets is diphyodont dentition.
Got it. And the final term is heterodont.
Well, hetero means different. We don't just have one uniform type of tooth.
Right, we aren't crocodiles.
Exactly.
Yeah.
A crocodile has row after row of the exact same pointy tooth. But humans have 4 very different functional types.
Incisors for cutting.
Canines for tearing.
Premolars for crushing.
And molars for grinding. Which perfectly brings us to the dental formula.
Let's talk about the dental formula. 2, 1, 2, 3. This is absolute bread and butter for the Neet exam. But where do students typically lose marks here?
Oh, they lose marks on the definition of the fraction itself. Students see 2, 1, 2, 3 over 2, 1, 2, 3, and they immediately think it means the entire upper jaw divided by the entire lower jaw.
Wait, it's not?
No, and that's the trap. It is exactly 1/2 of the upper jaw over 1/2 of the lower jaw.
Oh, I see. Because if you just add the numbers up, 2 + 1 + 2 + 3, You only get 8.
Right, and you don't have 8 teeth in your mouth. You have 8 in one single quadrant.
So to get the full mouth count, you have to multiply that fraction by 4.
Exactly. 8 times 4 gives you 32. That is your adult count.
And the order of those numbers is rigidly set. I always use the acronym ICPM to remember it.
That works. Incisors, canines, premolars, molars, 2, 1, 2, 3.
Now, here is a really tricky question I have seen pop up. What is the dental formula for a teenager? Or, phrased differently, a question specifically asking about the monophyodont teeth.
Ah, the one-set teeth. This is a brilliant deep cut. Remember earlier we said humans are diphyodont. We have 2 sets.
Yeah.
Well, the caveat is that not every single tooth is replaced.
Oh, right, because a kid's mouth is too small to hold all the adult teeth anyway.
Exactly. The initial milk teeth set lacks premolars entirely. There are 0 premolars in a baby's mouth.
Wow, okay.
And the very last molar, the wisdom tooth, that comes in much, much later in life. So those specific teeth come in once, and they never, ever get replaced.
Meaning they are monophyodont.
Precisely. The 12 permanent teeth that are monophyodont are your 8 premolars and your 4 wisdom teeth.
That is such a crucial distinction for the exam. Write that down if you're listening. Okay, let's move past the teeth. Let's look at the tongue.
Yeah. The tongue is a freely movable muscular organ. But the attachment point is the key anatomical feature here. It's attached to the floor of the oral cavity by a structure called the frenulum.
The little stringy part under your tongue?
Right. And then on the upper surface, you have those small projections, the bumps, called papillae.
And those are the taste buds, right?
But here's the nuance. The source text explicitly specifies that some of them bear taste buds. Not all of them.
Oh, really?
Yeah. A lot of students assume every single bump on the tongue is actively tasting their food. But many are just there to provide friction to help roll the food around.
That makes a lot of sense. So we chew the food, mix it with saliva, make a bolus, and we swallow. It goes into the pharynx, which is the common passage for both food and air.
Right, and then down into the esophagus.
But before we get down into the stomach, I really want to visualize the actual physical wall of this tube we're traveling down.
This histological structure is absolutely fundamental.
Yeah.
You take a cross-section of the alimentary canal, literally anywhere from the esophagus all the way down to the rectum, you're going to see 4 distinct anatomical layers.
I want to build a mental picture of this. Let's say I was a microscopic nanobot, and I decided to drill right through the side of your intestine, starting from the outside of your body, moving inward toward the food. What layers am I punching through?
Yeah, that's a great visualization. So you are coming from the outside, the abdominal cavity, and you're moving inward toward the lumen. The very first thing your drill hits is the serosa.
The serosa. I always think of it as the raincoat of the gut.
That's a decent way to put it.
Yeah.
It is the outermost protective skin. Technically speaking, it's made of mesothelium, which is just a very thin epithelium of visceral organs, along with some connective tissue.
It basically holds the tube together so it doesn't just dissolve or rub away against your other organs.
Exactly.
Okay, so our nanomot punches through the raincoat. What's layer 2?
Now you hit the muscle, the muscularis. This is the engine room of the whole system. This is what generates the peristaltic waves. But here is where the examiners love to set a trap.
The arrangement of the muscles.
Yes. You have to know which layer is on the inside and which is on the outside.
I always used to mess this up. Is the circular muscle on the outside or at the inside?
Think about squeezing a tube of toothpaste. You need to squeeze inward first to actually constrict the tube.
Right.
Right. That inward squeezing action is the inner circular layer. Then, once it's constricted, you need to push the contents down the line. That longitudinal pushing is the outer longitudinal layer.
Squeeze first, then push. Inner circular, outer longitudinal. I actually use the mnemonic ICOL, I-C-O-L. Inner circular, outer longitudinal.
ICL is perfect. Whatever helps it stick in your memory. But wait, there is a major exception to this rule.
The stomach?
The stomach. Because the stomach doesn't just push food along in a straight line, it churns it. It acts like a washing machine.
Right, so simple squeeze and push isn't enough?
Exactly. So the stomach has an extra layer, oblique muscle layer, and that allows for that dynamic diagonal churning motion to really mix the food with the acid.
Okay, so our nanobot is past the serosa, past the muscularis engine room. What is layer 3?
Layer 3 is the submucosa. This layer is formed of loose connective tissues. Think of it as the infrastructure layer.
Infrastructure meaning?
Meaning this is where you find the nerves, the blood vessels, and the lymph vessels.
Is there anything highly specific in the submucosa that students need to flag?
Yes, a very specific detail, and it is entirely localized to the duodenum.
Okay, what is it?
In the duodenum, glands are also present in the submucosa.
And those would be Brunner's glands, right?
Correct. Now, normally digestive glands are found strictly in the innermost layer, the mucosa. So finding them out here in the submucosa is a uniquely identifying feature of the duodenum.
So if an exam question shows a histological slide with glands hiding out in the submucosa, you know immediately, 100%, that you are looking at the duodenum.
You got it.
Okay, that brings us to layer 4, the innermost layer.
The mucosa. This is the lining of the lumen itself. This is the layer that physically touches the food you ate.
And it changes shape depending on where you are in the tube, right?
It does. In the stomach, it forms irregular folds called rugae. Just think of a crumpled up paper bag. It allows the stomach to expand massively when you eat a big meal.
But in the small intestine, it looks totally different.
Very different. In the small intestine, the mucosa forms these small, finger-like foldings called villi.
Let's really zoom in on those villi, because this is where the biological concept of form follows function really takes center stage.
Absolutely. The entire evolutionary goal here is to maximize surface area. You want maximum possible contact with the liquid food so you can absorb the nutrients efficiently.
Right.
So you have these villi sticking up, but it goes deeper. The individual cells that actually line those villi produce their own numerous microscopic projections.
The microvilli.
Yes. And because there are so many of them, it gives the cells what we call a brush border appearance. The source text explicitly states that this specific modification increases the surface area enormously.
And what is actually inside one of these villus fingers? It's not just a solid bump of tissue, is it?
No, not at all. It's a highly active logistics hub. Inside each villus, you have a dense network of capillaries to absorb sugars and amino acids, and you also have a very large lymph vessel called a lacteal.
A lacteal. And we are going to see exactly why that lacteal is so critical when we tackle the fat absorption problem later on.
Yes, that is a huge topic.
Before we leave the mucosa, are there any other features here?
Two things. First, the mucosal epithelium has goblet cells. They secrete mucus, which just helps lubricate the whole passage.
And the second?
The crypts of Lieberkühn.
Like caves.
Exactly like caves. These are deep invaginations or pits located right in between the bases of the villi and the intestine. So the mucosal landscape isn't flat at all. It's this incredibly complex terrain of high peaks, the villi, and deep valleys, the crypts.
Okay, so we have fully built the tube. We understand the hardware. Now let's look at the primary storage tank, the stomach.
The stomach is located in the upper left portion of your abdominal cavity. Anatomically, it is described as a J-shaped bag. And it has 4 major structural parts.
Wait, 4? I clearly recall older textbooks saying it only had 3.
That's right. Older editions did say 3. But the updated standards specify 4, and you must know all 4 for the current exams.
Okay, let's list them.
First, you have the cardiac portion. This is the region into which the esophagus directly opens.
Near the heart, hence cardiac.
Exactly. Then you have the fundic region. This is the top dome of the stomach, and it is usually just filled with gas or swallowed air.
Then the main body.
The body, which is the main central region where most of the mixing happens. And finally, the pyloric portion, which tapers down and opens into the first part of the small intestine.
And guarding the entry and the exit of this tank are the biological traffic controllers, the sphincters.
Yes. At the top, you have the gastroesophageal sphincter. It regulates the opening of the esophagus into the stomach. Its main job is to prevent acid from splashing back up.
It's acid reflux.
Right. And at the bottom, you have the pyloric sphincter. This guards the opening into the duodenum, ensuring that the acidic food paste only leaves the stomach when it is perfectly ready.
Now let's dive into the cellular level of the stomach. The gastric glands located in that mucosal layer we talked about. This is a very high probability topic for multiple choice questions. We have 3 major types of secretory cells here.
Let's start with the simplest one, the mucous neck cells. Just as the name suggests, they secrete mucus.
Which seems basic, but it's totally vital.
It is life or death for the organ. Without that thick mucus barrier, the stomach's own acid would literally digest the stomach wall. You'd get massive ulcers immediately.
Okay, cell type 2.
The peptic, or chief cells. These cells secrete a proenzyme called pepsinogen.
I want to flag that prefix right now. Proenzyme. That means it is completely inactive when it's secreted, right?
Correct. It is harmless in its current state.
Which brings us to the 3rd cell type, the heavy hitters.
The parietal or oxyntic cells. They secrete 2 very powerful things. First, hydrochloric acid, HCl.
We know what the HCl does. It drops the pH, creates the acid bath, and actually activates that sleeping pepsinogen.
Right. But the second thing the parietal cells secrete is arguably even more interesting: intrinsic factor.
Why is intrinsic factor explicitly listed as essential?
Because intrinsic factor is absolutely required for the absorption of vitamin B12 later on in the intestine.
Without it, you can't absorb B12 at all.
Not a drop. And that leads to a severe condition called pernicious anemia. So even though it's a digestive secretion happening in the stomach, its ultimate impact is entirely systemic.
Meaning you could eat all the iron and steak in the world, but if your parietal cells are damaged and you lack intrinsic factor, you're still going to be severely anemic.
Precisely. It is a brilliant example of systems biology.
Okay, we have the anatomy fully set. We have the tank built. Now let's flip the switch and actually run the machine. Let's trace the chemical cascade, starting from the very moment food enters your mouth.
So the buccal cavity performs two major functions: mastication of food, which is chewing, and facilitation of swallowing. But chemically, that digestive process is initiated right there by the hydrolytic action of an enzyme.
Salivary amylase, also known as pyelin.
Yes, it's a carbohydrate splitting enzyme.
What are the exact specs on this enzyme? What environment does it need?
Its optimum pH is exactly 6.8.
So it's slightly acidic, but still very close to neutral.
Right. And at that pH, it hydrolyzes starch into a disaccharide called maltose.
Now, does it digest all the starch in the bread you just ate?
No, not at all. It only handles about 30% of the total starch.
Why only 30%? Is the enzyme just weak?
No, it's just a matter of physics and time. The food simply doesn't stay in your mouth that long. You chew a few times and you swallow.
And once that bolus hits the stomach?
The pH immediately drops from 6.8 down to roughly 1.8.
Wow.
And that extreme acid instantly denatures the salivary amylase. It physically destroys the shape of the enzyme, so it completely stops working.
Oh, also, there's one more thing in saliva we shouldn't skip. Lysozyme.
Good catch. Lysozyme is present in saliva, and it acts as an antibacterial agent. It helps prevent infections from the food you eat.
So we swallow the bolus, it passes down the pharynx, through the esophagus via those muscular peristaltic waves, and it hits the stomach. Now we are in a truly hostile environment.
Very hostile. Like we said, the pH is 1.8. But that incredibly acidic pH is highly optimal for the stomach's own enzymes, the pepsins.
Let's trace that specific protein breakdown. The Chi cells release that inactive pepsinogen.
Right. And upon exposure to the hydrochloric acid, that pepsinogen gets converted into the fully active enzyme, pepsin.
And pepsin is proteolytic.
It is. It aggressively breaks down intact proteins into smaller fragments called proteoses and peptones.
Now, what about babies? Because they drink a lot of milk, and liquid milk would just wash right through the stomach, wouldn't it?
It would. But infants have a very specific enzyme adapted just for them. It's called Rennin. And note the spelling here. It is spelled with a double N, R-E-N-N-I-N.
Rennin.
It's a proteolytic enzyme found strictly in the gastric juice of infants. It helps in the digestion of milk proteins.
It actually curdles the milk, right? It turns it into a solid mass.
Exactly. By curdling the milk protein into casein, the food physically stays in the infant's stomach much longer. That gives the pepsin actual time to work on it.
And you said spell it with a double N, because there's a trap there.
The classic examiner trap. Do not confuse it with renin, spelled with a single N.
Single N renin is a hormone secreted by the kidneys, right? It regulates blood pressure.
Correct. Single N is for blood pressure. Double N is for baby digestion. Do not mix those up.
Is there any fat digestion happening in the stomach at all?
The text mentions that very small amounts of lipases are secreted by the gastric glands. But realistically, it is extremely minor compared to the massive fat digestion that happens in the next phase.
Which perfectly brings us to the small intestine, the ultimate mixing bowl of digestion. This is where the real magic happens.
It really is. The small intestine receives major secretions from three different sources. You get bile from the liver, you get pancreatic juice from the pancreas, and you get intestinal juice from the intestine itself.
Let's focus on the pancreatic juice first, because now we have reached probably the most high-stakes, dangerous part of the entire digestive system. The pancreas. I always visualize the pancreas as a loaded gun.
That is a remarkably apt analogy. Think about it. The pancreas manufactures incredibly powerful enzymes designed to completely destroy and digest solid protein.
Right.
But the pancreas itself is an organ made entirely out of protein.
So if those enzymes were accidentally activated while they were still inside the pancreas?
It would literally digest itself from the inside out. It's called autodigestion or acute pancreatitis.
Yeah.
It is excruciatingly painful and often fatal.
So evolution had to solve that problem.
And it did.
Mm-hmm.
The pancreas ships everything out in a strict safety lock mode. It only secretes proenzymes, or Zymogens.
Let's list the squad of these inactive zymogens.
You have trypsinogen, you
Yeah.
have chymotrypsinogen, and you have procarboxypeptidases. Notice those specific prefixes and suffixes.
The gen at the end and the pro at the beginning.
Right. In biology, those terms mean inactive. They are fast asleep, completely harmless.
So they travel safely down the pancreatic duct, utterly harmless, until they physically drop into the duodenum. Then what happens?
Then they encounter the detonator. The intestinal mucosa secretes a very specific enzyme called enterokinase.
Enterokinase. That is the spark.
It is the spark. Enterokinase essentially has one single job in life. It finds the inactive trypsinogen and it cuts off the safety lock. Boom. Now you suddenly have fully active trypsin.
And trypsin is basically the general of the army.
He is. The moment trypsin wakes up, he runs around and wakes up everybody else. He activates the chymotrypsinogen into chymotrypsin. He activates the procarboxypeptidases. It triggers a massive chain reaction.
It is genuinely brilliant engineering. The key to the weapon, the enterokinase, is kept in a completely separate physical room from the weapon itself.
Right.
So what is the final result of this coordinated pancreatic attack?
It's a total breakdown. The proteins, proteoses, and peptones are converted into dipeptides by trypsin, chymotrypsin, and carboxypeptidase.
Okay, that's the proteins. What about the carbs?
The complex polysaccharides, like the remaining starch, are hydrolyzed by pancreatic amylase into disaccharides.
Then the fats.
Fats are broken down by powerful pancreatic lipases into diglycerides and then further into monoglycerides.
And nucleic acids, like the DNA in the plant cells you ate.
Nucleic acids are broken down by nucleuses into nucleotides and then into nucleosides.
That covers the massive pancreatic contribution. Now let's talk about the second major fluid, bile.
Right. Bile is released into the duodenum from the gallbladder where it was stored. Now, bile contains several things. It contains bile pigments, specifically bilirubin and biliverdin. It contains bile salts, cholesterol, and phospholipids.
But there is a massive neon flashing sign fact here.
The critical fact that every student must memorize. Bile contains absolutely zero enzymes.
No enzymes at all. Then how does it actually digest anything?
It doesn't digest chemically, it digests physically.
Hmm?
Think of trying to wash a greasy, oily frying pan.
Water just beads up and rolls off.
Right, you can't wash it with just water, you need dish soap. Bile basically acts like a biological detergent. It facilitates the emulsification of fats.
Meaning it breaks big blobs into smaller blobs.
Exactly. It breaks down massive fat globules into incredibly tiny droplets called micelles. This astronomically increases the total surface area for the water-soluble lipases to actually get to work. Bile also physically activates those lipases.
Okay, so after the pancreas and the bile do their work, we have a soup of dipeptides, disaccharides, monoglycerides, and nucleotides. But we aren't quite at the absorbable stage yet, are we?
Not quite. We need the absolute final step, the last mile delivery. And this is carried out by the succus entericus, which is the intestinal juice itself.
The brush border enzymes?
Yes. These enzymes act on those end products we just listed to form the absolute simplest absorbable forms. And this all happens immediately adjacent to the mucosal epithelial cells.
Let's run through those final chemical equations.
Okay, dipeptides are broken down by dipeptidases into single amino acids.
Got it.
Maltose is broken down by maltase into 2 molecules of glucose.
The basic energy currency.
Lactose is broken down by lactase into 1 glucose and 1 galactose.
And sucrose.
Sucrose is broken down by sucrase into 1 glucose and 1 fructose.
What about the nucleotides?
Nucleotides are broken down by nucleotidases into nucleosides, which are then broken down by nucleosidases into raw sugars and bases.
And the fats.
The remaining dipylcanine and monoglycerides are broken down by lipases into free fatty acids and glycerol.
And just to clarify the anatomical geography for everyone, the entire final breakdown happens specifically in the duodenum.
Yes, the text specifies that this intense chemical breakdown occurs in the duodenum region. The resulting simple substances are then pushed along and absorbed in the later sections, the jejunum and the ileum.
Now, before we fully move on to that absorption logistics phase, we really need to quickly profile the specific accessory glands that manufactured all those juices, the liver and the pancreas.
Let's start with the liver. It is the single largest gland in the entire human body. It weighs about 1.2 to 1.5 kilograms in a normal adult.
And what is its internal architecture like?
Its core structural and functional units are called hepatic lobules. Inside these lobules, you have hepatic cells arranged in radiating cords.
And there is a very specific anatomical covering on these lobules, right?
Yes, each individual lobule is covered by a thin connective tissue sheath called Glisson's capsule. This is a highly specific identifying term for mammalian liver anatomy. If you see Glisson's capsule on a test, it's the liver.
What about the duct system? Tracing the plumbing of the liver and gallbladder is a classic diagram-based question.
It is. Okay, let's trace it. The hepatic cells secrete the bile. That bile passes through tiny hepatic ducts and flows out of the liver.
But it doesn't go straight to the intestine.
No, it is diverted to the gallbladder, where it is stored and highly concentrated. Now, when the gallbladder squeezes, the bile exits through the cystic duct.
Cystic duct?
Right, and that cystic duct joins together with the main hepatic duct coming from the liver. Together, they form the common bile duct.
And that common bile duct continues down until it hits the pancreas.
Exactly. The common bile duct joins with the main pancreatic duct. They merge, and they open together into the duodenum as the common hepatopancreatic duct.
And there's a final valve there, right?
Yes. That final shared opening is guarded by a valve called the sphincter of Oddi.
Sphincter of Oddi. Great name. Okay, and just to officially round out the pancreas itself.
We already covered its exocrine function, secreting the alkaline pancreatic juice with all those Zymogen enzymes. But remember, it is a compound organ. It has an endocrine portion, too.
The hormones.
Right. The endocrine portion secretes hormones, mainly insulin and glucagon, directly into the blood to regulate blood sugar levels. But for Chapter 16, we focus primarily on the exocrine side.
Okay, brilliant. So the chemistry set has done its job. We have fundamentally broken the food down into raw glucose, free amino acids, fatty acids, and bases. Now we enter the logistics phase. Absorption. How do we actually get this stuff into our body? Where does it happen?
Well, technically, absorption happens everywhere, from the mouth all the way down to the large intestine. But the vast majority occurs in the small intestine.
Let's do a quick organ-by-organ sweep to be thorough, starting with the mouth.
The mouth absorbs very little food, but certain drugs coming into contact with the mucosa of the mouth and the lower side of the tongue are absorbed directly into the blood capillaries lining them.
Oh, this is why sublingual medications work so incredibly fast. They entirely bypass the slow digestive tract and go straight into the bloodstream.
Exactly.
What about the stomach? Does it absorb anything?
It does. Absorption of water, simple sugars, and alcohol takes place in the stomach.
Which clearly explains why you feel the neurological effects of alcohol relatively quickly on an empty stomach.
Yes, it crosses over very fast.
Then we hit the small intestine.
The principal organ of absorption. This is where the heavy lifting happens. Glucose, fructose, fatty acids, glycerol, and amino acids are all actively absorbed through the mucosa into the bloodstream and the lymph.
And finally, the large intestine.
No significant digestive activity happens here at all. Its main functions are the final absorption of some water, certain minerals, and specific drugs. And, of course, the massive secretion of mucus to adhere the remaining waste particles together for easy passage.
Now let's talk mechanics. How do these microscopic nutrients actually get across that mucosal cell wall? We have three distinct physiological mechanisms.
First, you have simple diffusion. This process depends entirely on the concentration gradient. Small amounts of monosaccharides like glucose, some amino acids, and some electrolytes like chloride ions just drift across passively from high concentration to low concentration.
No energy required.
None. But it's slow.
So what's the second mechanism?
Facilitated transport. This is for substances that need physical help crossing the lipid membrane, either because they are too big or too polar. Certain amounts of glucose and amino acids use special carrier proteins to shuttle them across.
And again, no ATP energy spent.
Right. But the third mechanism is active transport. This occurs completely against the concentration gradient. You are forcing molecules into a space that is already crowded.
So that definitely requires energy.
Yes, it burns ATP. Nutrients like various amino acids, monosaccharides like glucose, and specifically electrolytes like sodium ions are absorbed this way.
Wait a second. You just mentioned glucose in all three of those categories. Simple diffusion, facilitated, and active.
I did, and it definitely creates confusion for students. But the text clarifies it. Very small amounts of glucose can move by simple diffusion. Some uses facilitated carriers. But active transport is the primary energy-dependent method for the bulk uptake of glucose, often coupled directly with sodium.
Okay, that clarifies it. Now we have to tackle the big one, the fat problem.
Oh, the fat problem. This is a massive concept.
Because fatty acids and glycerol are completely insoluble in water, and blood is mostly water. They can't just casually swim into the tiny blood capillaries like glucose does. They would clump up and block the vessels.
Right, it would be a disaster. So the body uses a crucial multi-step packaging process to solve this.
Let's break down the steps. Step 1.
Step 1 happens out in the lumen. The free fatty acids and glycerol are first incorporated into those very small droplets we talked about earlier.
The micelles. Emulsified by the bile salts.
Yes, the micelles. Because of their structure, these micelles are able to physically move into the intestinal mucosal cells. They slip right across the membrane.
So the micelle is basically just a microscopic ferry boat to get the fat inside the cell. But what happens once the fat is inside the cell?
That is step 2. Once inside the mucosal cells, the fats are completely repackaged. They are reformed into very small, specialized, protein-coated fat globules.
Protein-coated, like putting a water-soluble wrapper around a piece of fat.
Exactly. And these specialized protein-coated packages are called chylomicrons.
Chylomicrons. That is a must-know vocabulary word.
Absolutely. So now you have these chylomicrons. Step 3 is exporting them, but they are far too large to fit through the tiny pores of a standard blood capillary.
So where do they go?
They are transported out of the cell and dumped into the lymph vessels, specifically those large central lymph vessels inside the villi that we mentioned an hour ago.
The lacteals.
The lacteals. The chylomicrons enter the lacteals. These lymph vessels carry the milky white fat emulsion all the way up through the lymphatic system, and ultimately they dump the absorbed fats directly into the large veins near the heart.
So fats literally take the scenic route via the lymph system because they're simply too large and too hydrophobic to enter the microcapillaries directly.
Precisely. It's an incredible workaround.
All right, let's shift gears a bit. Let's talk about energy and control. We go through all this trouble to eat for energy. How do biologists actually measure that energy?
We measure it using the unit of a calorie or a joule. But there is a very important distinction in the text that you have to understand. The difference between gross calorific value and physiologic value.
Gross calorific value. That is what happens in a piece of lab equipment called a bomb calorimeter, right? You literally put the food in a metal chamber, fill it with pure oxygen, and burn it to ash to see how much heat it gives off.
Exactly. It's a measure of absolute total energy. But physiologic value is the actual amount of usable energy your body can extract from that same food. And they are not the same number.
Because our bodies aren't perfect incinerators, we're a little inefficient.
Right. Let's look at the numbers. For carbohydrates, the gross value is 4.1 kilocalories per gram. But the physiologic value is a flat 4.0.
Very close. We are good at processing carbs. What about proteins?
Here is a big difference. For proteins, the gross value in the lab is 5.65 kilocalories per gram, but the physiologic value in the human body is only 4.0.
Wow, that is a significant drop.
It is. Proteins have a lot of complex nitrogen bonds that our bodies have to spend energy to break down and excrete safely as urea. So the net energy yield is much lower.
And finally, fats.
For fats, the gross value is 9.45 kilocalories per gram, and the physiologic value is a massive 9.0.
Fats are the undisputed winner in pure energy density. 9 calories per gram compared to 4 for carbs and protein.
More than double. And that is exactly why the human body preferentially loves storing excess energy as fat tissue. It is just incredibly efficient biological packaging.
That makes no sense. Now, how is this entire massive factory controlled? We talked about the symphony earlier. Who is the conductor?
It is a dual control system, a mix of neural and hormonal controls. Neurologically, just the mere sight, smell, or even thought of food stimulates saliva and gastric secretions via the central nervous system.
The Pavlovian response.
Exactly. Local neural networks in the gut wall also constantly moderate the muscular peristaltic activities.
And hormonally?
Hormonally, local hormones produced directly by the gastric and intestinal mucosa travel through the blood to regulate the precise timing and volume of the digestive juices. It's a highly automated feedback loop.
We are entering the final stretch now, part 7, when the system fails. The disorders of the digestive system.
Well, the most incredibly common ailment is simply inflammation of the intestinal tract due to standard bacterial or viral infections, or parasitic infections like tapeworm, roundworm, threadworm, pinworm.
Which are very common globally. But the source text explicitly highlights a few specific physiological conditions we need to cover. Let's start with jaundice.
In jaundice, the liver is the primary organ affected. The classic symptom is that the skin and the whites of the eyes turn visibly yellow.
Because of the bile pigments.
Yes. The liver fails to process and excrete bilirubin properly, so this yellow pigment deposits into the tissues of the body.
Next is vomiting.
Vomiting is the forced ejection of stomach contents back up through the mouth. It is an involuntary reflex action. But the key anatomical fact to know is that it is controlled by the specific vomit center located in the medulla of the brain.
The medulla, got it. And the text notes that a distinct feeling of nausea almost always precedes vomiting.
Yes, nausea is the neurological warning sign from the medulla.
Then there is constipation.
In constipation, the feces are abnormally retained within the colon. The natural bowel movements become irregular and infrequent, leading to excessive water absorption and hardened stool.
But the disorder I really want to spend the most time on today is PEM, protein energy malnutrition. The textbook has an entire dedicated box on this, explicitly comparing marasmus and kwashiorkor.
Yes, and this isn't just dry biology. This is sociology. This is human history.
It really is. The text directly references the Liberation War in Bangladesh and the severe droughts in Ethiopia in the 1980s. We are talking about the physiology of extreme starvation.
Right. But scientifically speaking, starvation actually manifests physically in two incredibly different ways, depending entirely on what specific macronutrient you are missing.
Let's distinguish them carefully, because the examiners absolutely love to swap the symptoms in the multiple choice options to trick you. Let's start with marasmus.
Okay. Picture an infant, specifically a baby usually under 1 single year old. This child is receiving essentially nothing in their diet. No substantial food.
So they are completely deficient in both raw calories and protein.
Yes, a simultaneous deficiency of proteins and total calories. This typically happens if the mother's breast milk is replaced far too early by other foods which are incredibly poor in both protein and caloric value.
Like just watery gruel.
Exactly. And socially, this often occurs if the mother has a second pregnancy very quickly, when the older infant is still far too young to be weaned.
And what are the physiological symptoms of marasmus?
Extreme, devastating emaciation. The child experiences severe thinning of the limbs. The skin becomes dry, very thin, and severely wrinkled because the subcutaneous fat is completely gone. Body weight declines drastically, and crucially, growth rate and brain development are severely impaired.
Essentially, the body is cannibalizing itself to survive. Skin and bones is the visual hallmark of marasmus.
That's right, complete wasting away.
Okay, now carefully contrast that entire clinical picture with kwashiorkor. And I think the name itself is highly revealing here. It comes from a Ghanaian word, roughly, meaning the sickness the baby gets when the new baby comes.
That etymology tells you the exact mechanism. If a new baby is born, the older child, who's usually over 1 year old, gets abruptly weaned off the protein-rich breast milk.
But they aren't totally starved of food.
Right. They are usually put on a cheap, locally available, highly starchy, carbohydrate-heavy diet.
So they are actually getting calories. They have enough energy.
Exactly. But they are getting practically zero protein. Kwashiorkor is caused strictly by protein deficiency, unaccompanied by a calorie deficiency.
And this leads to a very confusing physiological symptom, edema, severe swelling.
It is incredibly counterintuitive if you don't understand the biochemistry. You see a starving child, but they look swollen.
Because they lack the blood proteins.
Yes. Without adequate dietary protein, the liver can't synthesize essential blood proteins like albumin. Albumin normally acts like a sponge, holding water inside the blood vessels via osmotic pressure.
So without that protein sponge, the water literally leaks out of the veins.
It leaks out of the vascular system and pools in the interstitial tissues of the body. So you see extensive edema, swollen limbs, and the classic protruding pot belly.
Even though their underlying muscles are completely wasting away, just like in marasmus.
Exactly. But the wasting is masked by the retained fluid. And unlike marasmus, they often retain some amount of subcutaneous fat because they're still eating carbohydrate calories.
So let's build the ultimate cheat code for the exam.
Okay.
Marasmus equals an infant under 1 year. It is a deficiency of calories and protein. And the symptom is dry extreme wasting, skin and bones.
Perfect.
And kwashiorkor equals a child over 1 year. It is a deficiency of protein only. And the defining symptom is extensive swelling and edema, the pot belly.
That is a flawless simplification for the exam. If you keep those contrasting points locked in your mind, you won't get tricked.
Okay, we have chewed, digested, absorbed, and even troubleshot the entire biological system. What is genuinely left?
Just assimilation and defecation. Assimilation is very simply the final cellular process where the absorbed substances finally reach the tissues and are actively utilized for their designated activities, generating energy, building muscle, repairing cells.
The sandwich finally becomes you.
Precisely. And defecation is the disposal phase. The unabsorbed, undigested digestive wastes solidify into coherent feces in the rectum.
Which triggers a signal?
Yes. The distension of the rectum initiates a neural reflex, causing an urge or desire for its removal. Defecation itself is a voluntary process, and it is carried out by a massive peristaltic movement of the colon and rectum.
So, stepping back, what does this all really mean? We started this entire hour with the philosophical idea of systems biology.
Just think about the sheer, staggering efficiency of the machine we just described. You have a single, continuous biological tube that seamlessly changes its internal pH from 6.8 in the mouth, drops it down to a flesh-melting 1.8 in the stomach, and then instantly neutralizes it back to around 7.8 in the intestine, just to accommodate different specific enzymes.
It's basically a highly automated chemical refinery.
And you have a massive internal surface area in that intestine,
Correct.
radically amplified by millions of villi and microvilli, evolved simply to process massive, constant amounts of molecular data.
It really is incredible when you pause and think about it. We usually just eat a sandwich while watching a video and we don't think twice about it.
But your subconscious body views that sandwich as a highly complex code, a code of interlocked proteins, dense fats, and locked-up carbs that desperately needs to be cracked, meticulously sorted, and ultimately assimilated into your own biomass.
The enzyme cascade in the pancreas alone is a perfect example of that emergent property we talked about.
Right. It's an elegant regulatory system entirely preventing fatal self-digestion while simultaneously enabling the massive explosive chemical breakdown of foreign food. No single enzyme can do that. It requires the whole system acting in concert.
Well, here's a final thought for you to chew on, pun completely intended. We talked extensively about the surface area of the small intestine being enormous due to the brush border.
Yeah.
If you were to somehow magically flatten out every single one of those microscopic villi and microvilli in your gut right now, some physiological estimates say it would cover the entire surface area of a tennis court.
It's hard to even picture that.
That is exactly how much literal interface you have with the outside world, folded up and packed right inside your belly.
And remember, that massive interface is highly selectively permeable. It acts as the ultimate biological gatekeeper, deciding exactly what is allowed to enter your blood and become part of you, and what stays out in the lumen to be discarded.
It is a remarkable piece of machinery. That brings us to the end of our deep dive into digestion and absorption. We really hope this detailed conversation helps you connect the facts and build that mental model for your Neet prep.
Just remember, don't just blindly memorize the isolated terms. Visualize the three-dimensional anatomy, actively trace the chemical pathways step by step, and always try to understand the evolutionary why behind the structure.
Absolutely. Thanks for listening, everyone. Keep studying hard, and we'll catch you on the next